Hello, everyone, and welcome to this new Gen webinar entitled Automated IR Spectroscopy for Analysis of Protein Biopharmaceuticals. This webinar was made possible through sponsorship from RedShiftBio. I'll be your host today. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which is currently in its 40th year, bringing you the latest information on the most cutting-edge tools and technology that propels the life sciences industry forward. Biotherapeutics constitute about 1/2 of new drug approvals over the past several years, and there's really no indication that the market for these types of drugs is slowing down. Creating these complex protein therapeutic molecules is no simple task, so their analysis is even more challenging. Thankfully, infrared or IR spectroscopy has long been recognized as a powerful biophysical tool for determining protein secondary structure and monitoring dynamic structural changes. This is important since structural analysis helps establish process and product knowledge, inform the impact of upstream and downstream conditions, identify stable formulations, monitor product stability, and assess product comparability when process improvements are implemented. Put into practice, characterization of secondary structure is paramount in determining the accuracy of biosimilar molecules. And today we're going to hear more about an automated IR technique that can aid in the characterization of protein secondary structure. Here to tell us more about MMS, or microfluidic modulation spectroscopy, are our two expert panelists. First, we'll briefly hear from Dr. David Sloan, the Director of Applications at RedShiftBio. David will tell us about the AQS3 Pro MMS system from RedShiftBio and how it can be an integral part of your process development systems when ramping up biotherapeutic production. David will then turn the presentation over to our main speaker for today's webinar, Dr. Lucy Liu, a principal scientist at Pfizer in the Mass Spectrometry and Biophysical Characterization Group. Lucy is responsible for product and process characterization using various biophysical and biochemical techniques. And today, she's going to provide us with some real-world data using ASQ3 MMS technology for the analysis of therapeutic monoclonal antibodies. Right after Lucy's presentation, we'll have our Q&A session, and I want to remind the audience to submit questions for Lucy and David so we can have a really nice, lively Q&A discussion. To send in a question, it's really simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. You can do this at any time during the presentation, so don't hesitate. Once that question pops in your head, type it in and send it off. All right, now let's get our webinar started. David, I'm going to turn the mic over to you.
Hi, my name is Dave Sloan, and I'm the Director of Applications at RedShiftBio. Today, I'll give a brief introduction to the AQS3 Pro system and microfluidic modulation infrared spectroscopy, sometimes called MMS or MMIR. After my introduction, the main speaker today is Lucy Liu from Pfizer. At the heart of MMS are high-quality results. Automation increases reproducibility and also decreases hands-on time by over 20-fold from traditional secondary structural techniques. MMS provides a greater than 30-fold improvement in sensitivity over traditional FTIR and a 5-fold increase in sensitivity over circular dichroism. This high sensitivity and reproducibility gives you the confidence you need to move forward with only the best potential drug candidates. The AQS3 Pro system was introduced in 2019 and has since seen rapid adoption by many pharmaceutical companies and CDMOs around the world. Additionally, there is a rapidly increasing number of peer-reviewed publications from these companies, validating the ease of use and utility for the structural data being generated by the MMS or MMIR technology. There are 4 levels to structural characterization: primary, tertiary, and quaternary structural characterization have high-quality platforms with sufficient levels of automation to allow for the sample throughput demands of today's biophysical research labs. Historical secondary structural methods, such as FTIR and CD, are low-throughput manual techniques without the adequate dynamic range required for today's drug development processes. Frequently, both FTIR and CD must be performed to cover the necessary dynamic range and sensitivity demanded by the drug development process. Microfluidic modulation infrared spectroscopy addresses these historical shortcomings of secondary structural analysis. Complete automation for sample processing, as well as data analysis, allows for the rapid analysis of large numbers of samples in 24 or 96 well plates, with minimal hands-on time required. The MMIR data also has great reproducibility and high sensitivity for detecting small changes in overall structure as well as individual higher-order structural elements. We're focused on secondary structure analysis. Why is secondary structure so important? You cannot have correct tertiary and quaternary structure without the correct secondary structure. Secondary structure can change based on ligand binding and protein-protein interactions. A drug's formulation can affect its secondary structure as well as its stability. By monitoring secondary structure, the researcher can understand what is happening during binding events, what is occurring when different formulation buffers are tested, can understand what conditions may lead to aggregation, and what buffers and excipients provide the greatest stability and shelf life to a biologic drug product. Secondary structure is the foundational element to understanding the molecule's overall structure and final biological function. There are 4 keys to the MMS technology. A high-powered quantum cascade laser which provides 100 times more power than is seen with FTIR. The AQS3 Pro system is completely automated with throughput to match the needs of the experiment and the demands of the lab. Advanced analytical software automates the data analysis and also provides clear results which can be understood by the novice biophysical scientist, yet has enough flexibility to allow the advanced researcher to tailor the analysis directly to the needs of their study. Lastly, we have the high-precision flow cell. The flow cell is the key to the fluidic modulation. The real-time background subtraction and the drift-free analysis, which generates the high-precision results, structural data we get with MMS. Let me go into a bit more detail on the flow cell. Here we have a picture of the flow cell. The flow cell modulates back and forth between sample and matched buffer. We collect data wave number by wave number from both the sample and the buffer to generate a differential absorbance spectra. The rapid modulation provides drift-free background subtracted data, which is highly reproducible and sensitive to very small changes in absorbance within the amide-1 band. First, we collect differential absorbance spectra. We then interpolate the data to generate the absolute spectrum. We then convert that to a second derivative. We invert and baseline the data. This then allows us to deconvolute and identify the individual structural elements and percentages which constitute the secondary structure for the protein being analyzed. MMS is an essential part of the modern biophysical toolkit, providing the sensitivity and precision required to understand a protein's secondary structure and to monitor small changes in secondary structure elements based on formulation, concentration, ligand binding, and many different stress testing parameters involved in accelerated and real-time stability studies. MMS fits alongside SEC, DSC, DLS, hydrogen deuterium exchange LCMS, and NMR as a critical tool in today's biophysical research environment. The key measurements performed by MMS are aggregation, quantitation, stability, structure, and similarity. These 5 basic measurements impact a wide range of applications across a very broad range of research areas, such as biologic drugs, vaccines, enzyme ligand binding studies, as well as AAV characterization and nucleic acid therapeutics. It's my pleasure to introduce Lucy Liu from Pfizer with her presentation of a detailed overview of microfluidic modulation infrared spectroscopy, MMIR. Thank you for your attention, and I'm happy to answer questions at the end of the webinar.
Thanks for the kind introduction. Hello, everyone. My topic today is automated high-throughput IR spectroscopy for secondary structure analysis of protein biopharmaceuticals. First, I'd like to set the stage by describing the scope of the biophysical characterization. I listed here how our analytical research and development group organized to apply MAP platform knowledge and the state-of-the-art analytical technology. I'm in a technology-focused group called mass spectrometry and biophysical characterization. We focus on confirming primary structure, glycosylation, disulfide bonds, degradation pathways, and the scope of the biophysical characterization are mainly to look at 2 quality attributes, protein aggregation and high-order structure for all the products of different modality. A variety of techniques are involved in biophysical characterization. What you are seeing here for high-order structure, the secondary structure quality attributes is characterized by 2 different spectroscopy methods, far UV-CD and infrared spectroscopy IR. The new technique, MMIR, is what I will be focusing on today. MMIR is the acronym for microfluidic modulation, IR spectroscopy, that I will be referring to throughout the rest of the talk. Correct high-order structure is important to biological function. Strategic alignment with CMC regulatory expectations require implementation of orthogonal, complementary, and even redundant biophysical technique. Biophysical testing list is formulated based on knowledge drawn from FDA and EMA recommendations as highlighted in the pink box, where you can see for high-order structure, additional state-of-the-art methods can be they recommend should be considered, and IR is mentioned there. The testing list also is based on industry's late-stage project-related publications and on our own past comparability work experience and subject matter expertise. That brings me to the overview of MMIR, which is essentially an automated MI1 IR system. Why? Because MMIR specifically determines protein MI1 band at 1720 to 1580 base number, can routinely test with 1.4 mil sample for triplicate at 1 mg per mil protein concentration with 96 well plate. It is a pre-optimized configuration with very minimum tuning requirement. And its end-to-end software is very convenient for data acquisition, analysis, and reporting. MMIR versus FTIR. So the cartoon at the bottom showing that the MI1 bonds vibrate in different ways of stretching of the carbonyl group or binding mode of the secondary amine NH group. FTIR and MMIR both techniques cover MI1. I wanted to point out here the vibrational frequency range, though between these 2 techniques are different. FTIR covers the whole mid-IR range, 4,000 to 400 base number, while MMIR is only specialized just for MI1 band. To give you an idea, so to give you an idea what their capabilities are, so it can tie into your specific study requirements over different IR band regions. Now, a few words about MMIR to understand why it is superior in its focused region, MI1 band. For us, from this right simplified block diagram on the right, you can see a tunable laser which probes the protein solution through a microfluidic cell. The expanded view, which shows the flashing of the two glows switching back and forth between reference and the sample. The two streams are rapidly modulated across the laser beam path, shown as the red solid dot here. To alternately measure at 1 Hertz frequency, meaning at every 1 second switch the stream once and measure for a second between the reference and the sample streams. So it can subtract the reference stream signals or the buffer blank signals continuously to produce a drift-free measurement. Why drift-free? Because it continuously refresh the instrument referencing almost in real time with your sample measurement. The laser in MMIR is a quantum cascade laser, which is brighter than conventional laser. Therefore, MMIR made room temperature detector possible. On the other hand, on FTIR, the measurements are typically taken on a 1 minute intervals. So slight shift on peak position and the variation of peak intensity from a laser from the last minute interval test on reference to the next minute test interval test on sample are commonly observed. So this peak position shift and variation of the peak intensity shift are commonly observed for FTIR. So that causes signal to noise ratio after the process the sample spectrum to deteriorate. But in comparison, the core feature of MMIR is that it solved baseline drift. So hopefully you heard a few things that are going to be useful going forward with this MMIR technique. It has emerging technology for analyzing HOS would find application in many areas to help increase process and product knowledge. And so here are the 4 major categories of topic I will be briefly covering to demonstrate MMIR's instrument design, performance, convenience, and lastly, case studies. I'll start off with the first topic, auto-referencing and the real-time buffer subtraction. I would say this is the most powerful feature of MMIR. What you are seeing here on the right top panel are the overlaid FTIR spectrum of a protein MAP-A in buffer, and the buffer, and the water. So three signal overlays. We already know MMIR only acquire the MI1 band signal shaded in yellow. Only this narrow region, therefore, the water reference band on the left, shaded in purple here, are not collected around 2100 bit number. So we also know to obtain a high quality spectrum for protein in aqueous solution in the middle panel, the spectrum atmospheric water and the water in protein solution must be adequately subtracted from the observed protein spectrum. And while introduced in traditional FTIR, this arrow noted water band in figure A, serving as the reference region to show whether correct water subtraction is accomplished. If the water subtraction is correct, this region should be flat, as shown in the figure B, for buffer subtracted spectrum of MAP-A. And in figure C, for the water subtracted excipient spectrum. Now, given this context, first, we wanted to validate the buffer subtraction method by MMIR, which does not include the use of water absorbance band around 21 wave number, 2100 wave number. So a crossover study was carried out to compare the results. The top figure overlaid second derivative FTIR spectrum of a map sample from 20 to 87 mg per ml, while the bottom figure shows respective MMIR spectrum of the same map at concentrations which can go as low as 0.5 mg per ml. What happens is both by visual inspection and the similarity score in the middle table, despite different method of water buffer subtraction, both methods showed comparable second derivative IR spectrum. These compelling results suggest auto-referencing and the real-time buffer subtraction enabled MMIR to work equally well at low protein concentrations as at high protein concentrations. Then, how about the sensitivity in detecting structural differences? 3 IgG1 molecules, map A, R, and P, they were designed to bind different therapeutic antigens. In this side-by-side assessment, the second derivative IR spectrum of the 3 maps were overlaid, and the secondary structure compositions were compared in the bar graph on the right. You can see, although the beta sheet band closely match across the 3 maps, as expected for IgG1 antibodies, there are some subtle structural differences distinctly reflected in the region of 1680 to 1660 bit number, indicating some turn structure uniqueness of each one. These results suggest the MMIR is capable of detecting minor structural differences among different maps, which belong to the same IgG subclass. We further evaluated the robustness of MMIR to demonstrate the qualitative consistency between MMIR runs on different days by different analysts in intermediate precision robustness study. The results demonstrate the superior repeatability and robustness of the method. Next, I'd like to dive a little deeper to system suitability test, SST. This is to ensure the method variability was minimized for a more precise qualitative assessment. Now, the SST I'm talking about is on top of a system check protocol, which is already in place, part of the system suitability by the system. Definitely needed in terms of like signal to noise, bit number accuracy, and electronics performance. What we wanted here is to establish the SST with a commercial standard protein. The SST with the standard is to ensure the system produces the same reference IR spectrum each time before analyzing the test samples, as well as produces the same secondary structure composition. So we started this effort by working with the vendor using their recommended commercial protein standard hue, the lysozyme from hen egg white. We found one directly dissolving the lyl cake of hue in water. We got what's shown on the left, the spectrum, the resulting spectrum, and HOS composition table. And when we checked the sigma product sheet of hue and found this way of sample prep is not correct because the presence of 10% acetate buffer, acetate salt in the lyl cake had been included in the sample solution, but was not accounted for by the buffer blank here, which is just pure water. Later, we showed by dialyzing hue in water after reconstitution, a buffer blank exactly matches the buffer composition in which the sample is prepared, and using the final dialysate as a reference buffer. The resulting spectrum then correctly reflected just pure protein's standard hue. And the HOS composition table now is correct as well. And the differences, for example, for the major structure element, the alpha helix structure, you can see the content can be off by 6%. So we wanted to bring this up for attention. Dialysis is required for the perfect buffer matching algorithm to perform accurate background correction here in MMIR. Another big progress this year was to upgrade everything to 96 well plates. We really appreciate the render of making this design change, which enabled us to accommodate a large comparability sample set with the control samples and the buffer blank. And you also have the option to put the hue sequence suitability test on the same plate with your test samples for comparability study. So in summary, SST by hue are something that has significantly helped in moving forward with routine comparability test. We have put together here a flow chart which makes things easier to navigate through with 3 steps in it. As you can see, the SST wizard defined in software can help you set up a sample set with the hue standards by obtaining the replicates of this standards to check instrument precision with similarity score. So together with the reference spectrum of hue and the secondary structure composition values, which would help colleagues to know everything's working for the method. We also looked into how MMIR utilized area of overlap spectrum similarity scoring technique. There are many such approaches for area of overlap, and what MMIR uses is the one developed by Kendrick. So I do not want to get into a great detail here, but I did reference the articles that describe its initial use or the subsequent improvements at the bottom. Finally, a quick run through of 2 case studies shown here on the spectrum from a comparability study on the top and a similarity study at the bottom. So in both cases, that appear visually to be qualitatively similar for the samples under comparison. In case for similarity test, we did the same similarity assessment using FTIR at 20 mg per ml as well. You can see the bottom figure A, MMIR, measures a similarity, measures a biosimilar and the innovator products at 1 mg per ml and achieved high quality spectrum with good signal to noise. Spectrum similarity assessment analysis was applied where the similarity score calculated by comparing the area of overlap, the similarity score is 99.1%. So the results indicate MMIR provide high confidence in the similarity assessment. Both case studies demonstrate it can be used as a powerful comparability and similarity assessment tool in the biotherapeutic industry. So in summary, MMIR can measure protein samples from high concentrations to very low concentrations and provide high quality comparable data across a wide concentration range. And our data indicates MMIR is a powerful protein characterization tool for secondary structure assessment, demonstrating high sensitivity and reproducibility, as well as a readout of discrete secondary structure elements. Similar to FAR-UVCD and FTIR, MMIR appear applicable to modalities beyond MAPS and has great potential to become the primary characterization tool to routinely elucidate and monitor the secondary structure product quality attribute. In today's fast-paced therapeutic lab, where each project team maintain very aggressive development timelines, easy of use, and a fast turnaround, are becoming more and more important. And as a high throughput automated IR instrument, MMIR is found to bring increased resolution, sensitivity, and efficacy for next level biotherapeutic analysis. So finally, I want to point out this is a multi-year collaborative effort, which started as early as 2015, when Redshift was still called Flow Laser back then. I'd like to thank all the colleagues at Pfizer and the collaborators from Redshift for their leadership, help, and support. And thank you very much for listening.
Lucy, wonderful presentation, some really nice data in the application of MMS and the ASQ3. And David, thank you for introducing the system and its benefits to us. We appreciate that. To the audience, the Q&A is going to begin in just a few moments. So if you have a question for Lucy or David, don't hesitate. Type it in, hit send. It's really simple. It's right there on the right-hand side of your screen under the Ask a Question tab. Just go ahead and type it in, and we'll get that Q&A session started in just a moment. Okay, looks like we have a bunch of questions for Lucy and David, so let's start the Q&A. Bear with us for just a moment as we make that transition into the Q&A session.
All right, everyone. Thanks for joining us for the Q&A session. We have a bunch of questions for our panelists here. The first one's going to go to Dave. Dave, one of our audience members would like to know how much optimization of the parameters is needed before measuring each sample or spectrum, and for each sample set?
Thanks. That's a great question. And one of the really nice things of the system is it's completely automated from really all standpoints, but especially from any optimizations or any parameters that need to be set by the system. So the system really takes care of all of that, and the user doesn't need to worry about any optimizations when they're looking at their different samples, different buffers, different molecules that they're wanting to measure. The system does its own optimization for ensuring that there's no carryover, for ensuring that nothing is dirty in the system. It does its own optimizations from a background standpoint. It does its own optimizations for different buffers and everything like that. So the whole process is taken care of, so the user doesn't need to worry about it at all. It's really a super simple and straightforward system from an operational and an analysis standpoint.
All right, thanks, Dave. Next question we have is for Lucy. Lucy, an audience member says that the auto-referencing and buffer subtraction is an interesting feature of the technique, but the description of how this works and why it's superior, if it is, to traditional baseline subtraction is unclear. So maybe you could describe that a little more?
Yes, that's a very good question. So the MMS technique, the key feature is that comparing to FTIR, see FTIR measurement typically take like 1 minute interval. So the slight shift on peak positions and variation of the peak intensity from normally you start with a buffer check signal collection, then followed by the next minute interval you test your sample. But during this time, the water vapor level can be shifted and causing the signal to noise ratio change. So, but for this technique, it really get over that drawback. So the two stream of your buffer and your sample like switch at 1 second interval, basically can be seen as a real-time buffer subtraction. That's a great improvement for signal to noise ratio, and you can see from our case studies. And so that's basically subtract your reference signals and buffer blank signals but continuously to produce a drift-free measurement.
All right, thank you, Lucy. We have another question for Dave. This comes from Vivek at Lonza, who wants to know, what is the impact of the presence of detergents in the protein sample during analysis by MMS?
That is another great question, and it really hits on one of the strengths of the MMS technology and the AQS3 system. Because there is that sample and reference or real-time buffer or background subtraction that occurs, we're really impervious to the presence of detergents and really many other commonly used or commonly seen excipients in protein formulations. So it really goes directly to the flexibility of the system and the ability of the MMS technology to really analyze any sample in its real-world buffer formulation under the conditions you want to look at it. You don't need to put it into a real simple buffer. You don't need to translate it into a different buffer background just for analysis. You can look at the structure in the buffer that you care about. You can look at the buffer under the conditions that you care about, under the conditions that are going to be relevant for the formulation, for the dosing, for whatever other studies you're looking to do on that molecule.
All right, thanks, Dave. Stick with me. We have another question for you. An audience member would like to know, is there any water vapor subtraction applied to the MMS spectra?
Oh, that's a great question. Someone really knows IR-Spectroscopy. We address that really on a couple of different levels. The system is based on a QCL or quantum cascade laser, and it's a tunable laser. So on the most basic level, we choose the wave numbers that are based to slightly avoid or miss the major water vapor lines. So we're not hitting right on those water vapor lines, so we avoid them to some extent. And the other part of it is we have really robust background subtraction just because of the MMS technology, because we're flowing back and forth from a fluidic standpoint between sample and buffer. We can subtract out any of those water vapor absorbances, any of those water vapor lines that are present. And to be honest, it goes back to the previous question as well about detergents. We also subtract out any absorbance from excipients or detergents as well. But we really revolutionize IR spectroscopy based on our ability to remove those water vapor lines or absorbances from any of the other excipients or things that might be present in your sample. Thanks for that question.
All right, thanks for that, Dave. Lucy, we have another question for you. An audience member would like to know, are there any case studies where the far UV CD failed, but MMS excels?
Yes, that's a very good question. So right now, we are in the intensive investigation of one of our products, which changed from the lyophilized form in a vial formation, in a vial presentation into injectable pen presentation, like a drug product presentation. Then the regulator is asking us, because in the pen formulation, there are two additional excipients. One is a preservative, the other is detergent. They want to know in the real pen formulation if the secondary structure of your protein stay the same, because these two additional excipient interferes with the far UV CD analysis. So we were not able to do the far UV CD in the true formulation. We have to remove these two interfering excipients to do the far UV CD, but the regulators really want to know in the presence of these two excipients what it looks like. So we did some feasibility study with MMS. It turned out to be very superior. So we were able to do the, as Dave said, in the true world formulation to extract out the secondary structure of the protein. So because of the buffer subtraction and like real-time background blanking, basically, we were able to fully remove these two excipients interference from the proteins. So the conclusion basically, we were able to show to the regulators that the active protein is unchanged from the drug substance to the two drug product presentations, vial versus the pen, confirming the formulation excipient differences did not impact this product's secondary structure. So we are very happy we have purchased this instrument.
All right, thank you for that, Lucy. Dave, back to you. An audience member would like to know, what are the upper and lower protein concentration limits for this technology?
That's a great question, and is one of the other features that differentiates us from other secondary structure, other techniques that are out there. We do cover a super wide dynamic range of protein concentrations. And a lot of that comes back to the fact that we are working with a quantum cascade laser, and we do have a lot more power that we're putting into the system. So it does give us a really wide dynamic range and a very large ability to detect really low levels of protein and secondary structure. But to answer the question, we go down to about 0.1 mgs per mil, and we've successfully gone up to about 200 mgs per mil. So we cover a really, really, really wide dynamic range when it comes to protein concentrations. And really, just to throw it out there, we're not limited to proteins. We look at a whole field of biomolecules as well. We've done some studies with nucleic acids. We've done some studies with AAVs. So there's a lot of things that we look at, but we have a really wide dynamic range from, the low or sub mg per ml range up to the hundreds of mg per ml range.
All right, thank you very much. Looks like we have one more question for you here, Dave. Someone would like to know, do you demo the system?
Yes, yeah, we do. There's two different ways that people can get exposure to the system. We do proof of concept studies in customers' labs. So if someone's really interested, if someone really wants to see it up close and personal in their lab, we do proof of concept studies on site with customers. We also have we'll call it a sample submission program where a customer can send us samples, and we'll run those samples in our applications lab just to be lower impact on site and also to be aware of any COVID restrictions that may be present at customer sites. So you can send us samples or talk to us, and we'll send us samples. We'll analyze those results, and then we'll just report the results back to you. So there's really two different ways that you can get some, if not actual hands-on experience, at least data-rich experience in terms of the way we analyze samples, the type of results that we generate, and what the data looks like, as well as the data analysis or software package that we use for generating the results.
All right, thank you for that, Dave. And with that, it looks like we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website as usual for up to a year. So if you missed it, you can watch it again, or feel free to forward any of the links to any of your friends and colleagues, which we always recommend. I'd like to thank Lucy and Dave again for their very informative presentations, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to RedShiftBio for sponsoring this webinar. Hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.